Okay, I think it's now good to start. Yeah, thank you everyone for coming tonight for the third series of the talk after Thanksgiving week. Today we have Elahe Shenasa, who's a fourth-year PhD candidate in Interdisciplinary Oncology Program at UBC. She's also one of the oldest greenies in college. Yeah, it's really nice to have greenies is a term for like resident in Queen College, and it's really nice to have my friends like having a talk about her work that she's been doing for the past couple of years or longer. Yeah, without further ado, I'll leave the stage to Elahe.
Thank you, Yunsak, for the nice introduction, and thank you all for gathering here to listen to my presentation. Really happy to see some old faces and happy to see some new people here. So if you don't know me, I'm Elahe Shenasa. I'm a PhD candidate in the Interdisciplinary Oncology Program at UBC. And today I'll be talking about immunotherapy, which is basically using the body's own immune system to fight against cancer. And we would like to discover together to see if immunotherapy is a cure to cancer or not. So before I start, you know that recently we all experienced the pandemics, and we witnessed so many patients that recovered quickly from COVID, while others recovered slowly from COVID. So we know that it was our immune system that was fighting against COVID and viruses, right? And the reason that some patients recovered quickly and some recovered slowly had something to do with the strength of their immune system. So this is in the area of infectious diseases, but the same thing can be said about cancer, right? Immune system can detect cancer and can fight against cancer. And this is something that might be obvious to all of you, well, obviously from the title of my presentation, right? But this was a question, actually, a few years ago. So before I explain why this was a question, I want to talk about cancer. So cancer cells are just like normal cells, but they're a little bit selfish. So they would like to replicate. They would like to replicate a lot, and they want to do it very quickly. And because they're doing it very quickly, they actually miss some steps that a normal cell would just follow to replicate. And because they're missing the steps, they are not really organized, they're like not really functional. And anyways, the point is that they originate from normal cells. And that's why scientists in like 19th century thought that cancer cells are too similar to the body's own cells, normal cells, to be recognized by the immune system. They knew that immune system can recognize and target viruses and bacteria because they're outsiders, but it cannot really target cancer cells because they're too similar to the body's immune cells. So we know that it's not true, but what I want you to imagine is that think that we are in the 19th century, we're scientists, and we absolutely believe that immune system has nothing to do with cancer cells. And we want to walk through history and listen to some stories that are going to change our view about immune cells and cancer cells interactions. Okay. So the first story. The first story is the Elizabeth Dashiell's, or as we know her by her nickname, Bessie's story. So Bessie was from a rich family. She was a 17-year-old. And she came back from her cross-country trip to New York, and that was the moment that she realized that her hand is actually very swollen and there's a lump on her hand. And that was concerning. So her family took her to the hospital, and that was where she was seen by Dr. Colley. So Dr. Colley at the time was a 28-year-old surgeon, fresh out of Harvard. He was very young, but he was very skilled and a great surgeon. So he performed multiple surgeries on Bessie's hand, and he realized that she had cancer. And at the time in 1890s, there wasn't many options available to treat cancer. So he decided to amputate her hand, and unfortunately that didn't work because it was too late, and the cancer had already spread throughout her body. So unfortunately that girl died. But this made a huge impact on that young surgeon, because he thought to himself that I can't really witness a patient of mine die in front of me and I'm not capable of doing anything. So there must be a better way to treat cancer, and I'm going to find it. So he went to the library and he dived into medical records and reports to see if he can find something, a better approach to treat cancer. And one of the reports actually captured his attention. This was a helpless case called Fred Stein. So Fred Stein was a German immigrant. Again, he went to New York hospital because he had a tumor on his cheek, and there they diagnosed him with a cancer, and he had the same cancer that Bessie had. And so the surgeon tried to remove the cancer, but it wasn't really successful. They did it 5 times. They couldn't remove the entire cancer, the entire tumor. And then, because he was undergoing so many operations, he was at a hospital, he got an infection, a really bad infection. So in 1880s, 2 things could definitely kill a patient. One, being diagnosed with cancer, with the same type of cancer that he had, and going through surgeries, and the doctor tells you that the surgery isn't successful. And second one is getting that type of an infection that he had. So he had both. So the surgeon told him that, I'm really sorry, I can't really do anything about you. Just go home and say goodbye to your family. And that's exactly what he did. He went home, and a year later, he came back to say hi to his doctor. So the doctor found that his tumor has disappeared. And that was very interesting. So, Coley was reading the report, and he thought that that's very interesting, but there must be a mistake. This is not really believable, because Bessie's surgery was successful. She had the same tumor and she died. Fred's surgery was not successful. He had infection and fever and he survived. So he decided to find it out himself. So he found Fred's address. He took a cab to downtown New York, and he knocked on every single door and said, "Hey, do you know Fred Stein?" And people were like, "No." They thought that he's crazy, but he wanted to really know what happened to Fred Stein, if he really exists or not. And while he was doing that, somebody opened the door and he had a scar on his cheek. That was Fred Stein. And this happened 6 years later. So Fred was happy, healthy, and cancer-free. So that was really interesting to Coley, and Coley just put these together and was like, there must be something going on, right? That patient with an infection and fever, his tumor was disappeared. So maybe it's the infection, maybe it's the fever that is actually shrinking the tumor. So again, he went back to medical records and he found another case. This one, this time he found a report that was published by this German surgeon called Busch. So Busch, throughout the course of his being a doctor, he found that some patients' tumors are disappearing by itself, and it's mostly happening after a high fever and infection. And this was something that people and healthcare providers were observing since like the ancient Egypt time, right? That tumors were disappearing by itself. And so this time this doctor decided to test this. So he waited for an opportunity, and the opportunity presented itself to him on a hot summer day when he had 2 patients visiting his clinic around the same time. One of them, he had an infection due to an injury, and his type of an infection was the type that he was looking for. And the other one was a 19-year-old girl with a tumor in the neck. And the tumor was so big, he said that it was like the size of a child's head. She couldn't breathe, she couldn't eat, she couldn't sleep. So, and she had the same tumor that Bessie had and Fred had. So he decided to test his hypothesis. And when I say test the hypothesis, he didn't take bacteria and cancer cells and go to the lab and put them together and watch them under the microscope to see how they fight against each other, right? It's 1868, where medical ethics didn't really exist. So he did something that a doctor in 2022 would never ever do. I guess you can guess. He infected the 19-year-old cancer patient with the infection that he got from the other patient. That is crazy, right? But it gets more crazy because her tumor disappeared after 2 weeks and she could eat, sleep, and breathe again. So Collie saw that and he thought that, oh, I should do my own experiment. I should see if that actually works. So he, while he was it was 4 months after Bess's death, and there was a patient with the same characteristic that I just described with all the other patients. He came to his clinic, and I think there are a few people who want to come in. I'm not sure if you want to join the presentation. I'll be forcing them. I guess they're now so embarrassed to join. Question? Yeah, you can. Oh, that's a good question. I didn't really memorize the name of the bacteria, but it was a type of a skin infection that was very popular back then in hospitals. It was very, very frequent, very, very prevalent in hospitals. Okay, so again, same thing that I described, that patient. We don't actually know his real name, but we call him Mr. Zola. Mr. Zola had a big cancer on his neck. He had underwent 2 surgeries, were unsuccessful. He had a few months to live, so he was like, yeah, Dr. Cole, you can do your experiment on me. And so, again, medical ethics didn't really exist, but it was better compared to 20 years ago, so the hospital at least said, well, it's a bad idea, don't do it. But, Collie said, okay, I'm going to drop my idea. He actually didn't. He went to a private department and did his experiment there. So he took Zola there, and he, again, infected Zola with infection, with the bacteria. And Zola first didn't develop fever, so he tried fresh bacteria, higher doses. He even called his student who was traveling in Europe and said, hey, I know a lab in Germany that has the deadliest bacteria, just get a sample and bring it to me. And that worked, yeah. Zola started developing fever, and as you can all expect, 2 weeks later, his tumor disappeared. So we know that, yes, there is a clear link between cancer shrinkage and fever, right? Because fever is indicative of an active immune response. It means that the immune system is active and ready to fight. And that's when the tumor disappears. So these were all successful cases, and he knew that if he wants to prove it to the medical community, he has to have more cases. So he started adding more cases to his clinical trial. So he started with 12 patients, all were treated in that private apartment. He infected them with bacteria, 8 of them developed fever, 6 of them showed partial remission. So partial remission means that the signs and symptoms of cancer is reduced, and full remission means that the signs and symptoms of cancer are fully gone. So 2 of them were cured, and 2 of them unfortunately died of an infection. So because 2 people died of the infection, he decided to, instead of using bacteria, he decided to use inactive bacteria, or the toxins from the bacteria. Because at this point he knew that this is not the bacteria that's killing cancer cells, it's the immune system. So if you can trigger the immune system without causing an infection, then the immune cells can kill the cancer cell. He didn't really know the process behind it, the pathways, things that we know these days, but the association was kind of obvious, right? So again, he added a lot more cases. He kind of experimented with toxins. He apparently developed 15 formulas for his toxins, and he was pretty popular back then. New York Times was writing about him in 1908 that he has a remedy of mixed toxins, it can cure cancer. But this was from the people, right? In scientific community, he actually received a major pushback. Nobody wanted to publish his results because he was a creative scientist, he was a great surgeon, but he was not a good researcher. He didn't keep track of what he was doing. He didn't keep track of his methodology, he wasn't following the detailed protocol, and he didn't have many cases. So they told him that the results aren't reproducible. So Um, Also at the time, he said that, okay, that's fine, give me enough funding, so I'll do my 5-year clinical trial, and I'll show you that this actually works. But they didn't. And at the time, there was someone called Dr. James Ewing, who's a pathologist and head of the New York hospital. He was working on radiotherapy at the time. And at that exact same time, he got $100,000 funding to purchase radiotherapy equipment, and which is equivalent to $2.4 million these days. So that kind of like changed shifted where the cancer therapy goes, right? So people started funding and focusing on radiotherapy and chemotherapy, and they pushed toxin, which we know was a form of immunotherapy, aside for 100 years. So that's why we have chemo and radiotherapy as a first line of treatment, but not immunotherapy, because it's something new, because people started revisiting the idea almost 100 years later. So unfortunately, Kali died a year later, but his idea didn't die with him because of his daughter. So his daughter actually went back, collected all these records, and she organized it, and she defended her father's ideas with the funding that she actually got from Bessie. You remember who Bessie was? The first girl. And from Bessie's family, actually. And she founded the Cancer Research Institute, which after 60, 70 years, it still works and still supports cancer immunotherapy ideas. So to wrap it up, these were all stories that taught us that an activated immune response, which is like fever, for example, a sign of an activated immune response. An activated immune system can fight against cancer and can kill cancer cells, right? So I have a question from you. If immune system is capable of targeting and detecting and attacking cancer cells, then why do we have cancer in the first place? You know, for example, I was reading this book that I'm going to show you later. They have a great example. It says that, why does my mirror detect cancer sooner than my immune system? Right? Patients go to hospitals and then they find, for example, they find a lump in their body without even developing a fever. Nothing. Like everything's fine until they go to the hospital and then there is a tumor in their body. What does immune system is doing in that case? If you are in the field and you know the answer, maybe not, but like, yeah. Anybody else? Can you guess like what is going on? Okay, I'll answer that. It's fine. So this time, to answer that question, I'm not going to tell you stories. We're just going to talk about science, because this again happened like it took people so many years, almost 90 years to find the pathways and mechanisms, right? So we actually know what's going on. So that is this. So you see a cancer cell, and then you see an immune cell over there, okay? So we know that cancer cells are expressing something on them, like a receptor. What's this? And immune cells are also expressing something on them that can detect that, like this. And once they detect each other, they have a connection. And that connection is like a signal that tells the immune system, the immune cell, that, hey, please get activated. So it activates the immune cell. And immune cell, once it's activated, it kills the cancer cell. This is the same thing that happens with like bacteria and viruses, right? The immune cell recognizes the cancer cell and attacks it. Now, kind of like explains what was the idea behind toxin, right? He didn't know at the time, but now we know that that was going on. But let's just talk about now the question that I asked: why do we have cancer in the first place? The cancer cells are very small. Yeah, they're very selfish, but they're also very smart. They evolved to express something else on the surface that recognizes something else on an immune cell that deactivates it. And that's why, yes, some immune cells detect cancer cells and they eliminate them, but then some cancer cells kind of escape and then they express something else that deactivates the immune cell. That's why they can really replicate, go everywhere throughout the body without being recognized. So the person who actually identified this mechanism and discovered it, describes this as a car and a brake. So an activated immune cell is an immune cell that is going very fast, right? So activated is going very fast. And that signal, the deactivation signal, is like a brake. It's like that car will stop and will not be activated, like it's deactivated and it stops. It's like a brake to the immune system. So they started calling these checkpoints. So checkpoints are just the signals that deactivates the immune system, the signal that is like a brake. It's the checkpoint. So that person who discovered that also realized, okay, so what if I block that connection? Then I can keep the immune cell activated, right? I can reactivate the immune cell, I can keep it activated. So blocking this can happen with like a protein, like a molecule or a protein, which the one that I want to talk about is kind of form of an antibody, but think about it as a protein that is called checkpoint inhibitors. So again, checkpoints are just those receptors that are identifying each other, and then once they identify each other, they act like a brake that deactivates the immune cell and stops it. And checkpoint inhibitor is the one that doesn't let this happen. So it keeps the immune cell activated, right? And checkpoint inhibitors are drugs that we pretty much have. There are different types of immunotherapies that are available, and checkpoint inhibitors are one of them. I'll talk about them in a little bit, but so the person who discovered this, two people were actually discovered this. They were James Allison and Tasuku Honjo, and they got a Nobel Prize for Physiology or Medicine in 2018 for their discovery of cancer therapy by inhibition of negative immune regulation. So negative immune regulation was the checkpoints, the brake. Inhibiting that was that molecule that didn't let this happen. And so they discovered a new way of cancer treatment by inhibiting that connection. And so again, what Coley's toxin was doing was actually kind of activating the immune system, okay, and not letting that connection happen, right? But it wasn't very targeted, right? Checkpoint inhibitors are targeted. We know what we are targeting. We know where we are putting the protein. But the difference is that in his case, he was activating the entire immune cells in the body, kind of blindly activating it against tumor. But this one, we know what we're doing, right? So, and just a little bit about James Allison. He's actually a very interesting character. He plays harmonica in a band, and all the people in his band are immunologists or oncologists, and he calls his band checkpoints. And he actually plays, he has shows after conferences that are about immunology and oncology. And yeah, one of my friends is actually going to this concert in fall in Boston. So yeah, very interesting person. Okay, so anyways, he discovered it, and let's go back to this. Does anybody know this person? Yeah? Who's this person? Yes, great. Okay. So this person is Jimmy Carter. Do you know what does he have to do anything with this presentation? Any guesses? What? Yeah, exactly. He had cancer a few years ago. So yeah. So he went to a hospital because he had a tumor in his liver. And then they operated, and then they took the tumor out, and then they tested it, and it was a melanoma. Anybody know what melanoma is? It's a skin cancer. Skin cancer in the liver. It is not good. Okay, it meant that he initially had a skin cancer, and that skin cancer started spreading throughout his body, and then going to his liver, and then forming another tumor. They also found other lesions on his brain. So at this point, cancer is easier to manage when it's at one spot, right? The primary tumor, they call it primary tumor. It's easier to manage because you know where it is, you do surgery, radiation, it's easier to manage. But when it spreads throughout the body, then it makes it challenging. It's not the end of the world, but it makes it challenging, right? And his case was challenging. So he received radiation or radiotherapy for lesions that were in his brain, but also 4 rounds of immunotherapy with checkpoint inhibitors. So exactly inhibited that connection, inhibited that break, reactivated his immune system. And yes, immunotherapy saved his life. And 4 months later, he said that my most recent MRI brain cancer did not reveal any signs of the original cancer spots, nor any new ones. And so he was cancer-free after that. So this checkpoint inhibitors, he's an example of one of the million people who underwent immunotherapy, and that number is increasing. So here I'm showing a graph that shows the patients in the United States that had cancer and they were eligible to receive immunotherapy. They received immunotherapy, and that's shown in the blue graph. And it compares them to the patients who actually responded to that immunotherapy. So first of all, let's appreciate that starting 2014, there was a jump in patients who were eligible for immunotherapy, because that was when immunotherapy became available in clinical trials and for public. And so there was a jump, and then as we see that more patients are becoming eligible because they're testing these immune checkpoint inhibitors on different cancer types, so more people are becoming eligible. And more people are responding to these treatments, right? The graph's still going up. And there was a report that was saying that they're showing a 20% decrease in mortality rate, cancer mortality rate these days. And we know that it's because of early detection, obviously targeted therapies, but immunotherapy plays a big role in that number, right? Treatment options are available for patients who were hopeless previously. So it's all good news, but I guess you can see where the problem is, right? The problem is the gap. Not all the patients are responding to immunotherapy. It's like Holly's toxin. You remember only 2 patients had full remission out of 12. Some patients actually don't. And so what should we do? Should we give all the patients immunotherapy and hoping that some patient would respond? That's not going to be an answer, because these medications actually have severe side effects that can be deadly. So you cannot give it to everybody. At the same time, you cannot also ignore it, because some patients are experiencing cure, are experiencing complete response, and they would otherwise never achieve that, right? It's buying people's time, and that's important and that's valuable. So what should we do? Anyone knows what should we do? Any ideas? Sorry, I stopped asking you. So we have to select. We have to select those patients who respond, select those patients who don't. So patients who don't, they can save the time and go receive other treatments. And patients who do, then they know that they will respond. That's important. So that's kind of like where my thesis starts. So researchers wanted to know why, right? The answer, why do some patients respond, why do some don't? And the answer was probably in the tumor itself. So we took the tumor to do experiments on it. So how do we do that? We actually go back and take the tumors from surgeries. So there is a method to preserve tumors, and that's by putting it in something like a candle. And that way you can preserve tumor for like almost 50 years. So we go back to clinical trials that like happened 50 years ago, and we take these like candles, and then we slice it with like kind of like cut a very thin slice, like almost 4 micrometer thick, kind of like transparent see-through of that. And then we stain it, because it's kind of, again, transparent. You can't really see anything under the microscope with that. So we stain it to see what we find. One of the most easiest, most conventional, standard methods and cheapest is to do this. So this is called hematoxylin and eosine stained or ethne stained. So this is purple and pink. Think about it as like old days where televisions were like black and white, right? This is purple and pink. Very confusing, but pathologists actually go through several years of training to actually interpret these images. And they told us that the one on top is cancer cell. We just trust them because it's too complicated. And so you can see the cancer cells in two different patients, two different samples. One of them has a high number of immune cells. So immune cells are actually, I don't know if you can see that. You can see these dots that are dark purple are immune cells, actually. You see all of them. These are immune cells. This is a patient that has a high number of immune cells compared to this patient that has none. So if you're going to choose one to give immunotherapy to, which one should we choose? Sorry, I promise I'm not going to ask you questions, but we're going to choose this one, right? Good. Exactly. Yeah, exactly. So you have immune cells already there that you can activate, right? This one has nothing to activate. So I wish the answer was that easy. I wish we could say, like, this patient has high immune cells, it's going to respond to immunotherapy, let's just give it to them. This is not like that. Well, we know that, yes, there is a positive association. Yes, they tend to respond better, but not all the time. And so that's where my thesis jumps in. And what we say is that these immune cells are actually different types. But if we want to very simplify that, they have a pro-tumor type, which actually help the tumor grow by suppressing other immune cells, telling other immune cells, hey, chill, like, don't attack, it's fine. And anti-tumor cells that are attacking cancer cells, right? So these are different. And obviously there's a difference between patients who have a tumor that is full of pro-tumor cells versus patients who have tumors that are full of a combination of both or full of the other one, right? These patients are going to have different outcomes, and we can't really study that using conventional methods or standard methods because they don't show it to us. So in my thesis, I'm exploring different technologies and new technologies to study the tumor microenvironment and kind of like get a better image of tumor and what's going on in there. So the next slide is going to be a pretty image of one of these examples. This is not my project because I haven't really presented them at a conference, so I can't really show it here. But it's an example, a good example that I found. So pretty much the same samples, right? They're just like stained differently. And so the cells that you're seeing, they're kind of like form a tumor nest in cyan. They are cancer cells, you see. And then we have checkpoints, right? So checkpoint number one, PD-1 is a type of checkpoint, and anti-PD-1 is an antibody against PD-1 that is available as a checkpoint inhibitor, and I think that's the one that Jimmy Carter got. LAG3 is another checkpoint, it's a newly discovered one, and TIM3 is another checkpoint. And then the one that you're seeing in orange, there are those killer immune cells that are attacking cancer cells. And the ones that you're seeing in red are kind of like, they call them big eaters, macrophages. So these are kind of like eat the debris, and sometimes they are pro-tumor, sometimes they're anti-tumor, but their presence is important. So here, let's take a look. So I'm finding an immune cell, killer immune cell that is in orange, like here. And you see that this one's like expressing a little bit of the magenta color, right? I know that like in our offices, when we want to analyze these images, we can turn on and off the channels. Don't have it here. So this one that expresses magenta, it shows that this immune cell is not activated because this is expressing that checkpoint that I told you is like a break to the immune cell, right? For example, this one's actually expressing two different checkpoints. So once you see that, you're like, oh, probably if you give that patient an anti-PD-1 antibody, one type of checkpoint is not going to work because they have the other checkpoint. maybe you should target this patient with 2 different types of checkpoints, right? Just by simply looking at that image. And yeah, some of them are fully activated. For example, some like you can find some like orange probably here that don't show any other colors around them. So anyways, I think that this gives us a way better example and image of the tumor microenvironment than the previous one, right? Another thing that I wanted to say is that I'm all talking about the immune checkpoints and checkpoint inhibitors, but these are not the only immunotherapy drugs that are available. We have CAR T cells, has nothing to do with CAR, it's a chimeric antigen receptor that they just use the abbreviation CAR, and then newer versions of CAR, they just like added a bunch of other stuff on it and features, and they called it TRUC. And they actually found a very long abbreviation to make sure that this actually makes sense. Let me see if I can find it. Yeah, it's the CAR T cells armed with immune stimulatory cytokines and T cell redirected for antigen unrestricted cytokine info initiated killing. So it's like they wanted it to sound like TRUC. But anyways, you have like CAR T cells, TRUC T cells, and different types of biospecifics, small molecules, different types of immunotherapies. Okay, so, and this one again, I just like put it for all of us to appreciate this beautiful images. So these are different types of cancers types that are stained with this panel that you see. So PD-1 is a checkpoint, CD3 is types of immune cells, and CD27 is for memory cells, and the rest are checkpoints, and this is tumor cells. We see that how different cells, different types of tumor look different under the microscope. And yeah, I was just telling Pedro that we describe them as small galaxies sometimes. And we know that a group of people who were studying cosmology gathered together with a group of people who were studying immunology and oncology, and they had an idea, and the idea was that let's treat these images from cancer as if there are images of the stars or the galaxy. And then they applied the same algorithms that they had onto the same algorithms that they had for the stars onto these images, and they got a pretty great ideas of like, they tried to see which patients respond and which patients don't, and that was just a new way of looking at it. And that was very interesting. So again, this is just like ignore this one, the circle one. Okay, this isn't supposed to show up now. Just like, let's focus on the other one. So the other one, just like great example that puts this H&E images, the conventional method, next to the new method. And you can kind of like appreciate how this, they call this multiplex IF. IF is immunofluorescent. So this how immunofluorescent images look much better than the H&Es, but they also give us way more information than the other one. And it might look easy, let's just like stain them with different colors. It's actually very challenging because you have to make sure that they don't cross-react. You're putting so many markers on the same slide. You have to make sure that you don't lose the antibodies, the antigens on the cancer cells. And it's pretty challenging, and I think that people have managed to put like 7 to 8 markers on the same slide, and there are some new technologies recently that came out that are putting like 40, but just like kind of like manipulating it and using kind of computer algorithms on top of that to add some like fake stains on it. But again, that's one thing. And then, yeah, now we can go to the circle one. I think that's the one that was, I don't know if anybody noticed, it was on my first slide. Oh, nice, okay. So I'm ending this with my first slide. So this is just fun. I call this sailing around the stroma. So stroma is actually this connected, stroma is actually this connective tissue that connects these cancer nests. So these are all cancer, all in green. Okay, the blue ones are nuclei. Every cell has a nucleus. So thought that these cancer nests are like islands, and this connective tissue that connects them are like oceans or water. And so I just like painted a boat on it, and I called it Sailing Around the Stroma. But just kind of like, just to appreciate how beautiful those images are, and they're very exciting to look at them. And so, another fun fact about this. So do you know how small the tissue that I used to take this image from is? So it's 0.6 millimeter in diameter. 0.5 millimeter in diameter. Very small. And the way, why do we have to use such small images is that, such small tissue is that when we go back to patient surgical samples, we actually punch a core, and we just take a punch and we put it on another kind of like a candle thing. We put them all together. So that way you can put like 300 patients on the same block as you would want to put one patient, right? And that is because the funding is limited. You know, it's just you can't really do experiments on every single patient's full tissue section. So that, and some researchers were saying that these 0.6 millimeter cores are enough to represent the tumor. So anyways, this is just a small punch of tumor under the microscope. And I think that's it. Yeah, thank you so much for listening.
Well, thank you very much for the talk, and we have like 30 minutes for questions.
Thank you so much for the wonderful and really colorful presentation. I really liked it. So I have two questions. The first one is about this checkpoints. I was wondering, like, if there's any research about like how, like, what is the origin of these checkpoints? I know, like, it's the exhibition, you know, it's the expression of the gene, but at the same time, I was wondering, like, if there's anything interesting about, like, why, like, these cancer cells have these kind of checkpoints that kind of deactivate, because it kind of sounds like, you know, like counterintuitive if I think about the immune system naively. So yeah, I was wondering about that, and I think I'll ask another question later.
Okay. Yeah. So you want to know pretty much why cancer cells express checkpoints, right? Or like what's the mechanism behind it? So it's kind of an evolution, right? So when cancer starts, when normal cells start to become cancerous, they don't have it. So immune cells recognize them and target them and eliminate them, right? But the thing is that in a community of cancer cells or normal cells that are becoming cancerous, some of them escape because they have that, right? Kind of an evolution or like selection, right? Natural selection. Those who have it escape, and because they have it, they replicate and they form the right? So yeah, the person who actually discovered that mechanism got a Nobel Prize as well.
You mentioned that some of these drugs, by activating the immune system, can generate other kinds of side effects as the immune system then begins to attack other parts of the body as well as the cancer cells. Could you say a little bit about that?
Yeah. So it pretty much has something to do with like how do you activate the immune system, right? Coli's toxin with bacteria would activate all the immune cells in the body, and they would just like all start to attack everything, right? The cancer cells and all the other organs in the body. And so these days, when they want to activate, they target the checkpoints actually, checkpoint inhibitors target specific checkpoints. For example, for the PD-1 that I showed one of the checkpoints, we know that it's mostly expressed on one specific type of immune cells. like it could be expressed in other cells, but one type of immune cells. So once you activate that, that type of immune cells become activated. But our body, our immune system has negative regulators, right? For example, when we have fever, our fever gets better. When we have an infection, it gets better. Because immune system starts reacting, and I told you that there are some other types of immune cells that they tell them chill, just like don't attack. They kind of like regulate each other. They activate each other and then deactivate each other at the end, after any infection, right? So with the checkpoint inhibitors, the thing happens that we know that this is expressed on some type of cancer cell, so we don't activate the whole system. We activate part of it. And yes, it can still cause some side effects in some patients through something that's called like a cytokine release syndrome, which is when immune cells are releasing cytokines, which is pretty much toxins that are killing cancer cells. So both of them are all together releasing cytokines, and that can be bad. But once they regulate it, then they kind of like try to make sure that it doesn't really affect the entire body. But in some patients, it would.
Thank you so much for a wonderful presentation. So sorry, this is more of a history question, but so scientists understood cancer as like a unified concept relating to like tumors and certain symptoms in the 19th century. How far does, like I know cancers have presented throughout human history, just when, do you know roughly when people were first cognizant of like a tumor and affiliated symptoms as, and like naming it as one thing?
Mm-hmm. Or when they realize that the disease that they're dealing with is like cancer, for example.
Yeah.
So it's an interesting question, actually. For a long time, people think that cancer didn't exist for a long time, right? But it actually always existed from the beginning. But the reason that they never caught it was that many years ago, people used to die soon, right? So they would die, and you know that like people would die sooner before developing the cancer, right? So that was the main one of the reasons. So they never really find it. The other reason is that we know through like CT scans, MRIs, we can find lesions that are not visible with eyes. But back then, when they didn't have those, they didn't really know what they're dealing with. And I know that I was reading a book many years ago. I could be wrong, but the way they realized that they were dealing with cancer was actually looking at the samples under the microscope. So I think that before a microscope, it was also really difficult to differentiate between different lumps in the body that are causing the patient to die. So yeah, I don't remember exactly when, but it was probably after.
Thank you so much for such an interesting presentation. This is the first time where I sit in a talk about cancer, and I feel that it's beautiful. So this is one, which is very remarkable. Second, I have a question about the prevalence of like the treatment in several geographical areas. So as far as I understand, most of the research that has been presented in the case are like affiliated with North America, and please correct me if I'm wrong. So is the treatment applied in different parts of the world, and is it recognized in different parts of the world as like a primary treatment or like one of the treatments that doctors would consider?
Unfortunately, I guess the answer is kind of no. It's kind of like a first world country treatment. Well, immunotherapy is still new, right? So not many people even in North America have access to it, right? They're still doing research, they're still making it available to more cancer patients. But yes, unfortunately, I guess from what I know is that it's a first world country treatment and not available anywhere else yet. And I think that a part of it is just has something to do with the cost that it puts on the healthcare system, right? It's an important one. It has to be funded, but it's very expensive as well.
In your slide, you showed the graph with people that responded efficiently with to the immunotherapy and the people that didn't. There is some research that looks at the epigenetic changes that happen in people that have cancer and people that do not have cancer. in your research or colleagues that you know have worked on detecting the changes in epigenetics on these two cohorts that have received the treatment?
Okay, good question. I can explain what epigenetic changes are. So genetic changes are mutation in the genes, right? Mutations that are causing cancer. Epigenetic changes are changes that are not mutations, but they kind of regulate how these genes are being translated into proteins, right? So you can't really find the mutation itself, but it's something that is happening around it. And I'm not working on epigenetic changes. I don't know if one of my colleagues here, I don't know if she does, but
I don't think or at least I'm not too sure about how far they've actually gone into it. But definitely, I think, at least in the cancer that, like Elahe is working on immunotherapy, I work with some drugs, and we do have colleagues who work on epigenetics. So at the Michael Smith Center at UBC, they have a huge center there. So they do look at a lot of samples through, but I think because we need a lot of, when we're looking at the kind of results that, so when the kind of data that Elahe showed is very nice, and she's like, she's talking about samples that we get from like 50 years ago. But when we're looking at, tissue that we need to look at epigenetic results, we need to use very specific tissue, like they have to be very large amounts of tissue, organ tissue. So I think and it requires a lot of research. Like it's fairly simple to get data on that requires a lot more. So I think maybe once we start actually getting more patients and more tumors out, we might get more representative data. But specifically speaking, I don't
Thank you. It was a very clear presentation. This was very nice. You talked quickly about the CAR T cells, right? And we have Notch on the campus, who is one of the two first companies who are developing immunotherapy, right? And the CAR T cells, they are engineered immuno cells, right? Which are able to, because they are engineered cells, maybe to do more than normal immune cells would do. So I've seen a video where all these immune cells were swarming around the cancer, and they couldn't really get through the cancer, because once they activated, the cancer has a certain layer around it. Are these engineered T cells, or also we had at first the question, how can we make this gap smaller, and maybe can we make it 80% for all? With engineered cells, we could maybe do this, right? But maybe not yet, because we are not that far in science. If we get our immune system to work, we may not be able to do this. But with these engineered cells, are we able to go through the tumor, or how far are we in this? And what is the effect of these engineered cells on the population we already have inside our immune system?
Great question. So these CAR T cells that I said, they're again type of immune cells that people actually took those. They're T cells, a type of immune cells. They took them out of the patient, and then they armed them with things that they would go and recognize the tumor cell. They would send them back to the patient. And they mostly work in non-solid tumors, for example, for blood cancer, because they circulate throughout the body and they can detect cancer, it can attack it. And as Marilyn mentioned, the solid tumors, they can't really infiltrate the solid tumor. And people are still trying CAR T cell therapy for solid tumors, but it's not as effective as the blood tumors, because they need to find mechanisms and ways to make it infiltrate into that solid tumor. However, I think that you also brought up a very important point that, yes, biomarkers are important to select the patients who will respond and those who don't, but there are also other ways to make those who don't respond, and that is by CAR T cells, or kind of like inducing like kind of like an active immune response in their bodies, so that they kind of like immune cells go to the tumor site and then they become present to be activated.
We have a last question on.
Okay, thank you for the beautiful presentation. My understanding is, as over time humans have changed. The way our body was structured, let's say 100 years ago, is different from the way we are now and probably in times to come. And if cancer cells are from the body itself, are we then ever going to close the gap? Because probably over time, the cancer cells may change due to the source of the body, because the body changes over time. So, are we ever going to? I don't know. Do you think, from the progress of your research on it, do you believe that at some point we shall find a proper cure for cancer, or no, or a proper way to handle the situation?
Great question. Yeah. So yes, cancer cells actually change. And that's why we actually believe that immunotherapy could work, because you need a drug that changes with cancer. And what changes with cancer? Immune system, right? But you have like targeted therapy, chemotherapies, other therapies that are effective, but they're one formula, right? Give it to the patient develops resistance, they don't work anymore. But immune system is so powerful and so capable of adapting to changes and changing, that we think that because cancer cells change, it's good to use immune cell system against cancer, because it has the capacity to change accordingly.
Hello. I just want to know if currently, is there any practical clinical way of determining who will respond and who won't?
Great question. Yeah. One of the immunotherapy checkpoint inhibitors that actually really worked was that they used a biomarker. So I can probably show you here that you remember that I said that one of these checkpoints are PD-1. So for patients to use, in order for them to use the anti-PD-1 checkpoint inhibitor, they check the level of PD-1. Very simple. So they kind of obtain a very simple staining of that one biomarker, and then for patients who had a high level of PD-1, higher than a threshold, they suggested that they can use immunotherapy. There's some patients, I know that there's some clinical trials that the criteria to be entered into that trial and receive immunotherapy is that they need to have a combination of different types of immune cells. And they're called tertiary lymphoid structures, but pretty much those structures that are like lymph nodes that are formed inside of different areas of tumor. And then they look for those structures, and whoever has it will be referred to immunotherapy. It also, what we say biomarkers, but a tumor type can also be a marker, right? For example, melanoma patients or non-small cell lung cancer patients, different types of cancers are eligible for immunotherapy, because we know that they have higher mutation rates, immune system are kind of more active, and they can recognize the cancer cells. So there is value in reactivating that.
Well, thank you very much for the talk again, and I think we have an announcement from our principal. Oh yeah, of course.
Really give a huge cheer for Elahe and Robert. There's a couple of things to say, because this is one of those weeks where we don't have just one resident member presenting in the coach house. Actually, this week we have three. Elahe has led off. Wednesday night, 8 o'clock, back here. Resident member and filmmaker in residence, John Hopkins, who many of you have met, is going to present her work in progress for her next film, Atomic Bamboozle. You read about this at the website and the videos. So that's here, 8 o'clock on Wednesday night. Come and find out what Jan really does and why it's so important. And then on Thursday afternoon at 5, our very own Steve Chignell, who's kind of a geographer, but not a regular geographer, because he's doing stuff in IRES. retired faculty member, a great distinction, a current geographer, former head of the geography department, and between the three of them, they're going to try to figure out, under pressure from a moderator, what geographers really do all day. This is the first in a series of events, discipline by discipline. It's going to be next month will be English, in the new year there'll be economics, nursing, if we get lucky, molecular biology, and indigenous studies. We've had an interdisciplinary oncologist this evening. We try to be as interdisciplinary as we can at Green College, but many of us have disciplinary connections, even if we've sort of broken out. But what do those disciplines really do, and what happens to them as they evolve over time? If you're curious about geography, and if you talk to geographers, some of them do think they actually run the world. Come on Tuesday evening and put your own hard questions and spread the word. Then come back next Monday evening after 8 o'clock, because I think those things confirmed.
Yeah, we have meeting next Monday.
That's meeting.
Yeah.
Right. But right now, this is